Thank you for the invitation. I'm going to discuss characterization of a low-cost meshes to assess suitability for hernia repair under technology appraisal regulations, effect of ethylene oxide sterilisation. I have no disclosure to mention. Mosquito nets have been around for a while and effectively used as a low-cost substitute for expensive commercial meshes in the developing countries. Regulatory barriers in the developed world has acted as a barrier for sterilisation process, one of those which has prevented the use of low-cost commercial meshes in the developed world. Ethylene oxide sterilisation is one of those methods which is approved at present to use for most of the medical devices in European Peninsula and United Kingdom. We aim to assess the effect of ethylene sterilisation on the mechanical properties and the biocompatibility of two low-cost meshes available in the United Kingdom using in vitro cultures with human dermal fibroblasts. In our initial data on the mechanical properties, we showed the break stress, the break strain was significantly lower in the low-cost meshes once they been exposed to ethylene oxide sterilisation. And there was a slight increase in the mesh stiffness once they exposed to the ethylene oxide sterilisation. We found no difference in the human dermal fibroblast viability at 72 hours between the control group and the ethylene oxide sterilisation group. We also performed an immunofluorescence study to look at the differences between the cell attachment and the proliferation of the cells. Looking at the control group, looking at the low-cost meshes exposed to the ethylene oxide sterilisation versus no exposure to ethylene oxide sterilisation, we found that ethylene oxide sterilisation has a very slight difference between the cell attachment and proliferation. It has statistically less significant difference once the cell proliferation and the cell attachment was looked at via immunofluorescence study. The clinical differences has yet to be determined on a larger scale study. In conclusion, we found no difference between the cytotoxicity and a biocompatibility test among the meshes which were exposed to the ethylene oxide sterilisation versus not exposed to ethylene oxide sterilisation. And these findings provide crucial information for the adoption of this frugal innovation beyond the developed world, opening a much possible innovation as a cost-effective solution for high-income countries. Our future studies are aiming to look forward to perform these studies on a larger scale with on animal models as well. And that would probably highlight the certainty of a possible use of low-cost meshes in the developed world for a hernia repair or a soft tissue reconstruction of various types. Thank you.